All right, this is Unit 3, Part 4. We're continuing with neurophysiology, the function of neurons. Now, up until now, we've spoken about the cells that are in the nervous system, supporting cells, and also neurons. Then we talked about neuron function, but I just want to talk a little bit more now about the interaction between neurons. Now, as I told you, neurons have axons, and they send impulses out if they reach a threshold in terms of their depolarization. In other words, going to a more positive charge. If they reach the threshold, they'll action potential is triggered, and the impulse goes down the axon. And the fibers, the nerve fibers themselves, the axons, are classified according to a number of different things, including diameter, the degree of myelination, and by speed of conduction. Now, once the impulse reaches the end of an axon, it will either meet another neuron, or it will have a connection to an effector cell or effector tissue like muscle. We're going to restrict ourselves here to what happens at the junction between a nerve cell or neuron axon and another neuron. So we're going to restrict ourselves to talking about this example here. So if the place where they meet the junction is called a synapse, and if the synapse is between two neurons, then the neurons are designated as follows: a presynaptic neuron, that's the one conducting the impulse along its axon towards that synapse or junction, and the postsynaptic neuron, the one that receives that signal and can transmit impulses away from the synapse, further away. So here in this diagram, actually, you see an example. This is here the axon. It's a myelinated axon of a neuron, and it's meeting with the cell body or soma of a neuron, this particular neuron here. And here's another example of an axon from some neuron. Up here, there's a neuron, and the axon comes down, and it meets the dendrites of, again, this neuron. And then the third example here is a neuron with its axon actually meeting another axon, and that can happen, but it's rare, and we won't talk about those examples. The most common examples that you find are the axosomatic, an axon meeting the soma or cell body of a neuron, and axodendritic synapses, an axon meeting the dendrites and forming a junction or synapse there. Now, and that's what's shown in this slide: axodendritic synapses or axosomatic synapses. And the other types we're not going to discuss, and they're not as common as these types: axodendritic, between an axon of neuron and dendrite of another, or between the axon of a neuron and the soma or cell body of another neuron. Now, there are two major other types of classifications that you can put synapses in, and that's the nature of the stimulation involved that generates the impulse. We have electrical synapses and we have chemical synapses. And for the most part, we'll be talking about chemical synapses. Electrical synapses are less common. They correspond to gap junctions that are found between other cell types, and they're involved in the central nervous system in a number of functions. I wouldn't worry too much about that. Let's move on to chemical synapses. Now, chemical synapses are specialized so that there's a release of neurotransmitter at the synapse and a reception of the neurotransmitter by the postsynaptic neuron. So the presynaptic neuron releases neurotransmitters and the postsynaptic neuron receives. Typically you have that kind of situation where the presynaptic neuron has its axon terminals at a synapse, and inside that presynaptic neuron, inside the axonal terminals, you'll have the synaptic vesicles that contain neurotransmitter. On the receptor side, which could be typically dendrites of a postsynaptic neuron or the cell body of a postsynaptic neuron, that's where you have the receptors. Now, the space in between the two, in between the presynaptic and the postsynaptic neurons, that space is called a synaptic cleft, and it is fluid filled with interstitial fluid. It prevents nerve impulses from passing immediately from one neuron to the next. There is a transmission process across that synaptic cleft, and it's a chemical event, not an electrical event. It's a chemical event, and this way it ensures unidirectional communication between neurons, so that there's no flow in the going retrograde. It's all unidirectional down the axon to the synapse, and then across the synapse to the next neuron. So this is really all about the communication system, essentially between neurons. And later, when we talk about muscle, we'll talk about the communication between a neuron and muscle. That kind of junction is called a neuromuscular junction, whereas here we're talking about synapses between neurons. Okay, here what you see in this diagram is the axon terminal, one of the buds at the end of an axon terminal, containing synaptic vesicles that have within them the neurotransmitter. Now, the way the process works is that if a action potential arrives coming down the axon, so an action potential is arriving, this nerve impulse arrives down the axon to the axon terminal, then that will trigger entry of calcium ions from the extracellular fluid into the axon terminal, triggering the synaptic vesicles to fuse with this presynaptic neuron membrane and empty their contents, their neurotransmitter contents, into the synaptic cleft. That diffuses, that neurotransmitter will diffuse across and bind to receptors. And in this case, at this kind of chemical synapse, what you have are receptors that can also act as channels. This is an example of an unstimulated closed, here I'll do it in a different color. This here is an example of a closed ion channel. This protein in the membrane of the postsynaptic neuron has neurotransmitter receptors and has the ability to bind a receptor, and that's exactly what's happening here, right there. You see binding, sorry, not bind a receptor. It has receptors, and it has the ability to bind its specific neurotransmitter. It's a receptor ligand interaction. The ligand is a neurotransmitter in this case. So with binding, and again here there's binding. So with binding, what happens is that the protein changes shape, and you get a channel opening, and you see that channel right there in the middle. And again here, right here in the middle of this, same thing here. So these channels open when the neurotransmitter binds to its specific receptor on that protein. In that way, ions flow in, and the membrane potential changes. And that's what's described here. Nerve impulses arriving at the axon terminals and calcium channels opening in the presynaptic neuron, and neurotransmitter gets released in the synaptic cleft by a process called exocytosis. And you get diffusion, crossing over of the neurotransmitter across the cleft, binding to specific receptors on the postsynaptic neuron, and then those receptors act as channels so that permeability will change and ions can move across. Now, channels are very specific. They could let through particular ions but not others. So it is possible to get an excitatory effect because the ion that comes across causes depolarization, moving towards more positive. It's allowing in positive ions, or it could have actually an inhibitory effect if the neurotransmitter opens, if it's an inhibitory type of signal, and the neurotransmitter opens channels that allow negatively charged ions to move across. That can also happen. And in that case, would become hyperpolarized and less likely that the neuron on the other side would become activated. Anyhow, let's stick for now to the excitatory kind of scenario. So here you have it, the neurotransmitter. This is a single shown in the square number 4 here. That's shown here in this diagram. What you have happening is that the neurotransmitter is binding to a receptor on the protein that's in the membrane of the postsynaptic neuron. This, in this case, is an excitatory type of event, and sodium ions will flow through the channel and into the cell. And the sodium channels flowing in, the sodium ions flowing in, the sodium ions are more positively charged and therefore cause a depolarization of the postsynaptic neuron on the postsynaptic side, and so it becomes less. It becomes depolarized, less negative or more positively charged. Now, that kind of interaction of the neurotransmitter with its receptor cannot be irreversible. It cannot be a permanent kind of reaction. Otherwise, you'd have these channels open all the time. It's only a temporary event, and then you get release and breakdown of the neurotransmitter, so that impulses do not have a permanent effect. It's just temporary. And once the neurotransmitter gets released, the ion channel closes. Here you can see the channel in the center is now closed, and sodium ions can no longer easily flow into the cell. The neurotransmitter that binds will not produce a continuous postsynaptic effect. Once it's bound, you have an effect. I don't like this word continuous here. It really should be removed. It produces I'm going to remove it like this. It produces a postsynaptic effect, and it blocks any additional signals from coming in, but it must be removed. Binding is always reversible. So, and the removal or destruction of the signal is due to enzymes that are present within the synaptic cleft. So there are enzymes there that can break down the neurotransmitter, and it'll either, then the breakdown products get reabsorbed by other cells around, or by the presynaptic neuron, or it'll just diffuse away from the synaptic cleft. Now, whenever you have transmission from one neuron to another across a synapse, you have a delay, and the delay is due to the time it takes for neurotransmitter to be released, diffuse across, and bind to the specific receptors. And that kind of synaptic delay is typically anywhere from 0.3 to 5 milliseconds. This is a rate-limiting step in neural transmission, and allows for changes to occur, and more regulatory mechanisms to have an effect. Because of the synapse, you don't have direct communication. There is this process that's going on, and it can be affected to limit, when necessary, transmission. If it wasn't for that mechanism, then transmission would be immediate, direct, and it would not be good. Okay, so now let's talk about the postsynaptic side. What's happening on the postsynaptic, in postsynaptic neuron? The neurotransmitter receptors that are coming across are causing changes in membrane potential on the postsynaptic side, and that depends on how much neurotransmitter is getting released into the synapse and the amount of time that the neurotransmitter stays bound to the receptors. As I mentioned, you can have excitatory or inhibitory effects. It really depends on the neurotransmitter or the receptor or the receptors that are present. Excitatory effects result in a depolarizing effect on the postsynaptic side, and inhibitory effects result in hyperpolarization. So these type of effects on the postsynaptic side are called excitatory postsynaptic potentials, EPSPs, in which case you have, and I'll write it here, which cause, in this case, EPSPs cause a depolarizing effect. It's not very easy writing this. Depolarizing effect on the postsynaptic side. Or you can have excitatory postsynaptic potentials, which cause a hyperpolarizing effect. So it's hyperpolarizing. Remember, depolarizing refers to going towards more positive membrane potential, and hyperpolarizing means going towards a more negative. Hyperpolarizing. Effect. Okay, and in terms of excitatory postsynaptic potentials, these are graded potentials. They're small. They're not action potentials, but if there is enough of them happening, then they can trigger an action potential to occur at the axon hillock and head down the axon. Remember, you have to. It's a question of, will the axon hillock, the base of the axon, become depolarized enough to trigger an action potential that will then swiftly move down the axon of that neuron. So EPSPs are graded potentials causing a depolarizing effect. They use chemically gated channels and cause a flow in opposite directions of sodium and potassium at the same time. At the postsynaptic membrane, you don't get the action. You don't get an action potential. You get a depolarization, and if the sum total of all the depolarizations are enough, they will spread to the axon hillock, and there at the base of the axon, at the axon hillock, an action potential will be generated. Here you see the effect of a EPSP, excitatory postsynaptic potential, causing a small depolarization, not enough to hit threshold, and it has to spread towards the axon, and enough of them have to arrive at the axon to that combined together they have enough of a depolarizing effect that they'll hit threshold and trigger a action potential. In terms of inhibitory postsynaptic potentials, or at inhibitory synapses, neurotransmitter is binding to a receptor that has changes that can cause changes in permeability. Now, there's channels that open that are permeable to potassium and chloride ions. Potassium will move out because there's more potassium inside the cell than outside. So potassium leaks out, causing the cell membrane to become hyperpolarized, or chloride ions will move in, causing more, also causing hyperpolarization, going towards the more negative. Positives out and or negatives in. So that leaves the charge on the postsynaptic membrane as more negative. It does have a hyperpolarizing effect. Overall, the effect is that the postsynaptic neuron's ability to produce an action potential will be reduced because of that hyperpolarization. And here you see the hyperpolarizing effect of an IPSP, inhibitory postsynaptic potential. So what you get, in fact, happening in a neuron that's receiving signals will be the sum total of all the different kinds of signals coming in. How many signals are coming in, how often they're coming in, and whether they're excitatory or inhibitory or both. And if it's both, then which one is greater than the other? In order to generate an action potential at the axon hillock, at the base of the axon on the postsynaptic neuron, you have to have enough EPSPs that they summate together to induce an action potential. And summation can either be due to time, it can be temporal summation, or it can be spatial summation. So either temporal or spatial, and I'll explain in the next slide what that means. As I mentioned, if you have IPSPs, inhibitory postsynaptic potentials, they can summate with the excitatory and would cancel each other out. If enough IPSPs are coming in, inhibitory signals, then it could cancel the excitatory postsynaptic potentials. Now, in terms of summation, as I said, you can have temporal summation, in which case you have the presynaptic neurons are transmitting impulses very, very rapidly. Action potentials are firing rapidly, so you have a rapid fire kind of arrival of impulses down an axon. And in that case, you could generate an action potential, or it could be spatial summation. So you can have temporal summation or spatial summation. With temporal, it's rapid fire in time. With spatial, you have a number of neurons getting stimulated. You have a number of presynaptic neurons stimulating the postsynaptic neuron. So a large number of axon terminals at the same time are stimulating the postsynaptic neuron. So this is an example of temporal summation. Here you have a axon terminal that happens to be joining here, in this case E1, in this case on the upper left. You have an axon coming down. It happens to be joining onto the cell body of the postsynaptic neuron. And you're getting firing off here. There's one EPSP, and then there's a time delay in another EPSP, excitatory postsynaptic potential. But together, they're not close enough to summate to bring the membrane potential to the threshold. So there's no summation, no action potential generated. But if the firing is close enough, there's one firing, there's another one, if it's close enough in time, you get temporal summation, hit the threshold there, and an action potential gets generated at the axon hillock. Remember that it's always generated here. You have to get at the axon hillock, you have to have enough depolarization spreading to that area that it can trigger an action potential. In terms of spatial summation, you have more than one axon terminal or more than one presynaptic neuron sending its axons onto the postsynaptic neuron, and you get them both firing at the same time so that it reaches here, threshold, and an action potential gets generated at the axon hillock, resulting in this postsynaptic neuron now firing off a action potential down its axon. The summation between excitatory and inhibitory postsynaptic potentials is shown in the example on the right-hand side of the slide. Here you have an inhibitory neuron's axon coming down, and here an excitatory neuron's axon coming down, and the result of the firing off of the inhibitory one is a hyperpolarization, and if they both fire, the inhibitory and the excitatory, as shown here, they cancel each other out. Okay? All right. And this is a summation of more information. I wouldn't hold you responsible. You could have a look at it and read it. It talks about some of the things that I've been mentioning, but gives extra information. Okay, now it's important to realize that there are many different types of neurotransmitters used in the nervous system for communication. There are at least 50 different ones that have been identified, and they can be classified either chemically or functionally. The neurotransmitters, the chemical neurotransmitters, the one that is best described is acetylcholine, and we talked perhaps a little bit about it. It's abbreviated capital A, capital CH. There are a number of other neurotransmitter types that are mentioned in the next few slides, but I wouldn't hold you responsible for that. We're going to mainly focus on acetylcholine. So as I said, it was the first identified neurotransmitter. It's the best understood. It's the neurotransmitter that gets released at neuromuscular junctions, the junction between a neuron and the axon of a neuron and muscle tissue. It's the neurotransmitter. Acetylcholine is the neurotransmitter that's enclosed in the synaptic vesicles at the end of the axon terminal. And it, as I mentioned, the interaction between neurotransmitter and its receptor is not permanent. It is temporary. It gets reversed because of the action of an enzyme. And in the case of the neuromuscular junction, the enzyme is called acetylcholinesterase. It is and it's abbreviated like this, ACHE. Acetylcholinesterase will break down the neurotransmitter acetylcholine as it comes off of its receptor. It does get released from the receptor and will get broken down. All neurons that stimulate skeletal muscle at a neuromuscular junction release acetylcholine. And some neurons in another nervous system that we haven't discussed yet, the autonomic nervous system, also release acetylcholine. So it's quite a commonly occurring neurotransmitter, both for muscle tissue excitation or impulses to skeletal muscle, And also it's important in the autonomic nervous system synapses. And we'll talk about that in the last part of this series of lectures on the nervous system. And here's more information. Again, I wouldn't hold you responsible for what's here, but it's more information about the neurotransmitter acetylcholine. The other neurotransmitters, there was quite a list of them. We will talk later about norepinephrine and epinephrine, but again, I'm not going to hold you responsible right now for the information I'm showing here. But we will talk more about norepinephrine and epinephrine when we talk about the autonomic nervous system, and we'll cover more about acetylcholine also when we talk about that autonomic nervous system. So all of this here, I'm not going to hold you responsible for. There are many different types of neurotransmitters, some excitatory, some inhibitory, quite a complicated system. Okay, on slide 26, actually, I mentioned that we can classify neurotransmitters either chemically or functionally, and I've gone through some of the chemicals. At least I talked about acetylcholine and that there are others. We can also functionally classify neurotransmitters into either excitatory or inhibitory types. And of course, as I've talked about before, the excitatory neurotransmitters will cause depolarizations, and the inhibitory ones will cause hyperpolarizations. Depolarizations make generation of an action potential more likely, and hyperpolarizations will make a generation of action potential less likely. The inhibitory or excitatory effects also depend not only on the neurotransmitter, but also depend on the receptor type that's found on the postsynaptic neuron, the specific receptors for those neurotransmitters. Some neurotransmitters have multiple potential receptors that they could bind to, and if the receptor on the other side, on the postsynaptic side, is an inhibitory type, then you'll get a inhibition, hyperpolarizing effect, or if it's excitatory, you'll get a depolarizing. An example of a single neurotransmitter that has different types of receptors would be acetylcholine. At the neuromuscular junction, the junction between a neuron and skeletal muscle tissue, you always have an excitatory kind of effect. Acetylcholine will bind to receptors that trigger a depolarizing excitatory effect. However, if you're looking at cardiac muscle in the heart, acetylcholine acts on receptors that have an inhibitory effect. Release of acetylcholine will cause inhibition of the cardiac muscle and slowing down of its contractions. So you can see in this way that it's not just the neurotransmitter, but also the receptor type. You can have different neurotransmitters having inhibitory or excitatory effects, or you can have a single type of neurotransmitter which can have either excitatory or inhibitory effects depending on what type of receptor it is binding to. And receptors will differ in on different tissue types. So the other thing, of course, is the other thing is the neurotransmitters, as I've shown in previous slides, can bind to a protein that's acting not only as a receptor but as an ion channel, and will open the ion channel. And the example I showed you was acetylcholine binding at a synapse, binding, getting released by the presynaptic neuron, drifting across or diffusing across and binding to the postsynaptic neuron, to receptors which are specific and open a channel the protein opens up a channel and lets sodium ions go in. So that's a excitatory kind of direct neurotransmitter effect that directly binds and opens channels. Okay, so that's acetylcholine. But you can also have indirect effects where the neurotransmitter acts through what's called a second messenger. It binds to a specific receptor on the surface, but there's no channel necessarily that will open up. It will have effect on the inner side of the cell. I'm not going to get into the details of that, but it's an indirect type of stimulation. Here you see an example of a channel-linked receptor, acetylcholine binding to specific receptors and opening up this channel that's right in the middle here, opening, and the arrow shows that ions are flowing in. So this is composed of a integral membrane protein, which has the receptor and has the ability to open a channel. So that's a direct neurotransmitter effect. Its action is very immediate, very brief, because it gets reversed with release of the neurotransmitter and degradation or breaking down of the transmitter by enzymes. The system involves a ligand receptor interaction. The ligand in this case is the acetylcholine, and the receptor is the acetylcholine receptor, and ions will enter. If it's the kind of tissue that has excitatory receptors, you'll get depolarization of the membrane, and if they're inhibitory receptors, you'll get hyperpolarization. And there's the same diagram shown again, only larger. G-link proteins are examples of indirect activation, and indirect activation these effects are slower, much more prolonged, have longer action. They're involving transmembrane protein complexes that are present but are not able to form channels. What they do is they change in shape so that they have an effect on the interior side of the cell, and that's how they mediate their effect. So direct or indirect. Direct is faster, involves a neurotransmitter binding receptor and a channel opening, and indirect is slower and involves changes on the inside of the cell. And that is the end of Unit 3, Part 4.